I'm extremely excited to be here, not only because I'm hearing about all the wonderful things you guys are doing, but also because it took a while to get here. Bernie, I looked at my email, and our first interaction was January of 2016. So it took us a while to actually complete the contract and be where we are today. So I'm ecstatic. So I'm presenting iSITE. This is a new name for our platform. It was voted on last week on our social media. So what iSITE stands for is Integrated System for IBD Data Exchange. So it's obviously a LabKey server-based platform for consolidation of research data. The research data we focus on is IBD, which stands for Inflammatory Bowel Disease. This is described as conditions that are chronic inflammation of the GI tract. So there's two types of IBD, Crohn's disease and ulcerative colitis. In 2015, about 3 million individuals reported having IBD in the United States. And Mount Sinai has a close history with Crohn's disease because 3 of our physicians actually described the disease. Dr. Crohn, Dr. Ginsberg, and Dr. Oppenheimer. Obviously, because of just the last names, Dr. Crohn the disease was named after him just because he was first author, just due to order. So there are many contributors and users of the system, primarily researchers, clinicians, clinical research coordinators, anybody that's involved in a study. And the goal is to integrate all of the data with minimal disruption to each study workflow. So what does IBD research look like at Mount Sinai? Well, there are a ton of studies, and these are only 10 of them. They all focus on different perspectives or aspects of IBD, general registries to high-risk families, recently diagnosed individuals, and each one is run independently, managed by someone by themselves, so they're pretty much independent silos. And each one has their own consent permissions, data structure, language, ID assignment system, storage, so you can imagine it causes quite a bit of issues. So we have duplication. Each study, a person can be recruited multiple times in one study or the other because they do not crosstalk. Compatibility. Since each study has their own structure, talking from one study or trying to integrate cross studies is pretty much impossible. Very few fields overlap. Accessibility. Each place has its own storage, location. And in a study, they can store their data in 3 or 4 separate places. So it's a lot of places to access. And data integration not only across each study, but within the study. So integrating assays from the Immunocore with REDCap recruitment data or consent information within a study is also very difficult. So we're looking to LabKey to solve all our problems. But LabKey did have a lot of modules and factors that would really address all these issues. So this is what we asked LabKey to do. This was the simplified version of what I tried to sell them. So we have 4 main components that we're trying to combine and integrate: study data, which is everything collected from the study. And so this is recruitment, consent, and questionnaire data. Then we have the clinical data that's exported from our HR system, Epic. Specimen data, this is located in our cores or our pathology department, as well as laboratories. These are usually in FreezerPro or FreezerWorks structure. And big data, our omics, so SNP chips, sequencing, single-cell sequencing, RNA sequencing in general, CyTOF, a whole lot going on. So this is how we expect all the pieces to come together. Here in this box is LabKey. And I just wanted there are a lot of arrows. I don't want you to get lost. But the most important things I want to point out are that each study has its own folder. Each study wanted to have its own place where they could put their information and it would be familiar to what they're used to seeing. And then we have a separate integrated folder where we transform the information and allow everyone to see a similar structure. We pretty much enter all the information into the study folders and then through LabKey perform ETL to put it into the integrated folder. I'll just quickly go through all the pieces and how they get into the system. We'll start with study data. Study data is separated into registration information, which contains all of your identifiers, and then consent and questionnaire information. So consent and questionnaire goes directly into the study folders, whereas the registration information with the patient identifiers goes into a separate location. This is then run against an open EMPI system. This is a master patient index to identify duplicates. So this is how we get rid of our duplicates, which then feeds back into the rest of our system. So we make sure that when we integrate later on that we have identified who's the same. Then let's go to the clinical data. Clinical data, from the registration information, we get all of the MRNs that are associated with our patients. We hand it over to our clinical team, and then they shoot back structured EHR data that has been, our Institute of Personalized Medicine actually came up with these beautiful tables that we could incorporate into our LabKey system. Specimen data, this is, as I mentioned before, from many different sources. It comes in directly into our study folders, as well as big data. There will be some QC steps, as mentioned before from other groups, to decrease the size of and only upload the most reasonable variants of interest or relevant assays. And this is how we would like the pieces to come together. So I'm just going to go through each piece and tell you where we stand currently today. We, as I mentioned before, just downloaded the LabKey server, so we're still inputting information and getting familiar with the system. So we've obtained recruitment data from all 10 data sources. We've been able to run them through the EMPI and create unique identifiers. I've listed the relevant fields so you can get a sense of what we're collecting from each individual. Typical identifiers: first, last name, date of birth, sex, street address, email, race. Race is actually something we use in our algorithm, and MRN if it's available. Not everyone that's recruited is a Mount Sinai patient. And from this, we've generated wonderful templates to ensure that new studies that join our system know exactly what we're looking for so that we're not missing any of this information. So this is what, with LabKey's help, we were able to actually see recruitment across all of our studies in one place. This was amazing. We've been trying to do this since 2005, and it's finally happened. So we were able to see the overlap between all of our studies. There's a total of 7,066 patients enrolled. On the X and Y axis, we have all of the studies, and then you can see their overlap. If you go across the diagonal, it's the recruitment of each study. But the total unique study participants is only 6,457. This means that a little less than 10% of individuals are recruited more than once. So in order to be more efficient, we might want to focus more on newly recruited individuals than those that we already have samples for, since most of our studies are genetics-based. If we already have DNA on an individual and we already have the sample, there really is no need to constantly recruit them and ask them for a purple top EDTA tube and stop them from their clinical visit. So these are the number of studies in which research participants are registered. And you can see there's one individual that really likes to participate in our studies. So yeah, there's a large range of individuals that are recruited more than twice. So for our questionnaire, we have imported the raw data into the study-specific folders, and now we're currently working on the ETL process. This is really an interesting task, and you can see by the number of fields for each study, there's a wide range in structure and questions that each one asks. So RTP, for example, has a little less than 4,500 fields, whereas some of the smaller studies, such as the family study, only has 92. There's 154 fields that we are performing ETL on to query in the integrated folder. The field topics include everything that's disease-specific, since we're really interested in IBD. So disease activity, family history, medications, smoking, those are the factors that we've really pulled from these larger sets. And again, we've have a REDCap template questionnaire as a guideline for future studies. And the reason why we're using REDCap, just to emphasize, is because we wanted to do the current workflow to make sure that people would use our system. We didn't want to disrupt their usual work habits. So this is an overview. So for the first time, we were able to see what we actually have available to us. Studies talk, we meet at these conferences, but we really didn't know what we had at Mount Sinai for the IBD recruitment. And we were able to see that we've done a fairly good job of recruiting a diverse group. About half have CD, a quarter UC, and a quarter control, which is a really good distribution, especially for our genetic studies. And then for race, predominantly white, but this is what we traditionally see in IBD. As for consents, the integrated database adheres to all the conditions that participants agreed to per study. So this took also a lot of effort. 25 different consents were reviewed, and our consents at Mount Sinai are around 20 pages each, making sure that legally we had the appropriate flags for each individual so that only those that consented to being integrated would be. And from this, we realized there's a strong need for an omnibus consent and electronic consent, as all of these were paper copies. For clinical data, for those that consented to access to their EHR, we contacted Institute of Personalized Medicine, and they helped us in generating these 24 tables that we retrieved from our Epic system. They cover everything from encounters, orders, family history, medications, everything that our gastroenterologists believed were relevant to our studies. These 24 tables are what will be integrated with all our study questionnaires. I'm interested to see how they differ and compare those. It will be one of my preliminary analyses once we have everything in our integrated folder. The majority of participants do consent to EHR access, but since we recruit all over the nation and even internationally, only about 3,000 of them have clinical encounters at Mount Sinai, but it's still a significant amount. We also have, we were able to see the wide variety of specimens that are available. We haven't even incorporated FreezerWorks, which is one of our LIMS systems, and we already have a little under 30,000 samples. That's a lot of freezer space in New York City. So we have quite a few samples to play around with, and this is extremely exciting, especially for our residents and our future MDs that are looking for projects. So this is a large portion of this is not utilized. So it'll be a great resource for future projects. The integrated system will provide not all of the information about samples, but just what samples are available and where they're, the permissions and the freezer location. As for all the omics, this is the final piece, and this is the one that's going to need the most work, in my opinion. Right now, what we have is just a table stating what has been performed for each participant. Have they been sent for an exome chip or for CyTOF, or do they have single cell sequencing data? The next step will then be to work with our bioinformaticians and our techs and figure out what data is most relevant to be entered. We have to obviously standardize and have a QC and STEP. So I'm happy to talk about that with all of you that have gone through this. Next projects in our pipeline are the GSA chip array. We're sending the majority of our recruits across all studies to be GSA chipped, so we're going to have a ton of data. And we just sent off about 800 samples for whole exome seq. And this is the first big pull we did using LabKey. So it was great. I was able to do the pull very quickly. Instead of contacting 8 or 10 different people and trying to get all that information, I was able to make the manifest myself very quickly. What we learned. Finally, we know what's available to us. So this has been something that our entire team has wanted to know for quite some time, just because of the turnover and all the different studies and all the different people involved. We finally have a grasp on what's available. There's definitely a need for change in regulatory, an omnibus all-encompassing consent that we can all use, guidelines for protocols and workflows, and electronic consents. No more manual entry. That took weeks and weeks of work. So LabKey is really helping us to change our entire recruitment workflow at Mount Sinai. Templates are necessary. It doesn't necessarily need to be REDCap. We went with that because that's what our CRCs, or clinical research coordinators, are used to. And there's a high demand for collaboration platform. I'm already getting requests twice a week to use LabKey and have access to LabKey, so especially from different departments, which is always exciting. Future steps is to fully integrate Open EMPI. Right now it's done externally, and we pump out the results into LabKey. Patient lookup function in order to, as an individual is being recruited, check against our EMPI. Assay integration, as I went into detail in a prior slide, especially with genetic data. Since I'm part of the genetic and genomic sciences, this is priority. But single-cell sequencing is also one of, since it's a hot topic now, is of great interest. And one of the #1 requests from all the gastroenterologists was incorporating NLP and images. So many images and notes are kind of discarded just because they're difficult to shift through. So especially from pathology, radiology, and there's extreme interest in discharge summaries. So with the help of LabKey, we're able to put together the puzzle pieces and create a big picture, which helps us to utilize all of the resources that we have available to us, know those resources, and perform efficient research. And I would like to thank just some of the people that helped me with this. You can imagine with 10 studies and 3 more on the way, the team's much larger. But these are my go-tos, the people that keep me going, especially Tom, who's here, a software developer. He helps me with everything I can't understand. And Bernie and Adam, you've been very helpful. Thank you.